So at G.CLIPS, we believe actually that one protein can be several different targets based on the conformation that it can adopt, but also on the environment in which this protein is. So basically, if you want to have a specific candidate for a specific indication and with a specific effect, you have to choose wisely, actually, your material, your starting material and your antigen to have the conformation and environment that mimics as close as possible your target in the pathology of interest. And this is, of course, something that we have been studying for years now, and we know that for membrane protein environment is really important, and it can shape the structure and function of these proteins. We know, for example, that for some GPCR, the ions can really play a very important role in their function. We know as well that lipids can act as positive or negative allosteric modulators. So the lipidic composition of a specific cell type is very important in the function and structure of these protein. We know as well that in a pathological context, let's take, for example, the tumor microenvironment, we know that there's specificity such as pH that can play a very important role on the recognition between a ligand and the protein and the target of interest. And this is why it's really important to take this into account when we do our development and when we select for the candidate of interest. So at G.CLIPS, we realized, as many of you has seen, of course, I guess, that we have really some problems that we need to mitigate whenever we are working with this complex protein for antibody discovery. The first hurdle would be that we have difficulties stabilizing membrane proteins, and this makes them less accessible to work with in vitro. But also we know that this protein can adopt many different conformation. It can be just conformation, active state, inactive state, or others, but it also can be different formats, oligomers, monomers, dimers. And what is also important to know is that these proteins, since they are in different kind of cell types and organs, the tissue that we want to target need to be represented whenever we do our selection. And if it's not the case, if we don't have the conformation and the tissue of interest, this would lead probably to lack of selectivity, sorry. And finally, one thing that I really believe is important is that the fact that when we select for binders, we never know what they would do. And for me, it's really important, since the first steps when you start characterizing our binders and our hits, to kind of understand a minimum, what is this specific candidate doing to our target? Is it activating it? Is it inactivating it? It's an inhibitor, allosteric modulator. This is important because this will shape actually your selection for the lead candidate in the future.